In this lecture, we'll be taking a look at the role of personal identification in the work of not only forensic anthropologists, but criminal justice as well. Putting names to the unidentified begins with the development of biological profiles. So we have looked at the assessment of pathological conditions and trauma. There are additional considerations that must be taken when taking a look at the decedent and, in this case, unidentified remains, being able to determine a time since death, reconstructing perimortem and then antemortem events as well, and then developing what we'll be looking at in this lecture, which is a profile of the decedent based upon a variety of characteristics and evidences. The identification or the ID of unknown decedents is probably the most important aspect of forensic casework. Every unidentified body entering the medical legal system represents a missing person. Delays in identification can prolong things like family suffering. They can, in a sense, postpone legal issues with regard to not only developing a criminal case, but closing that case out. And then, of course, we need identification with regard to death certificates, notification of next of kin, the execution of wills, and then obtaining death benefits and estate privileges based upon survivors. There are a variety of methods that are used to establish identification. These include things like DNA analysis, visual recognition of the remains themselves, and then, of course, bringing in next of kin to be able to do that as well. Radiography, there's a variety of applications there. Probably the most common is dental radiography. Fingerprinting, and then a variety of biometric analyses, which also include our osteometrics and craniometrics. One very important consideration to make when gathering this kind of evidence and amassing it as part of your case work, you have to determine the decompositional state of the remains, the availability of antemortem data, and what you can say or can't say regarding the decedent. And then you have to consider admissibility. Admissibility in this case in terms of the court system, the judicial system, and the various aspects of jurisdiction as it relates to the work of law enforcement, and of course as that relates to your own work. Now, regardless of the methods that you are employing in the field, all personal identification of deceased individuals relies on pattern recognition. Visual identification by next of kin, fingerprints, radiographic comparisons, and DNA all involve the interpretation of patterns that you are witnessing, observing, recording in the field. In the case of visual recognition, patterns exist within the memory of people. They exist within antemortem photographs. With all other systems, post-mortem information and patterns are compared against things that we know with regard to populations and social data. There are databases that are maintained, and these are wonderful resources for law enforcement. Identifications can be made anecdotally through visual recognition of the decedent. There are circumstantial kinds of assessments that can be made with regard to associated artifacts. And of course, there are scientifically verifiable evidences, including DNA and fingerprint analysis. Whatever the case, identifications are based on either the existence of a unique identifier or the collective and consistent agreement of multiple features. So you're never looking at just one particular kind of resource when you're making an identification. It is, in a sense, a statement of probability based on the frequency of a suite of characteristics within a given population, and in this case, a human population. For example, likelihood ratios have been generated for genetic profiles within specific geographic regions. Another example, many of the osteometrics and craniometrics that we apply in the lab are things that are based upon not only the species, but then there are racial, ethnic, and geographical components to how we um, apply and utilize those measurements and assessments. We have to understand something about the context, even time, if you're dealing with archaeological material versus modern material. So likelihood ratios are things that really serve the work of forensic specialists very, very well. Understanding something about, in this case, mass casualties and populations. When you think about probability statements as they relate to things like dental formulas and direct X-ray comparisons, these are also things that, again, are pattern driven. We have to be able to identify particular kinds of anatomical and physiological features in what we are looking at, and then we compare those across groups. And this gives us better accuracy in terms of being able to assess this material in relation to one's identity. So the entire process of assessing the identity of a decedent must be considered a progressive process. We are including material, we are excluding material, we are looking at what could be, and we are definitely eliminating what is not probable. The goal here is to narrow the sample of potential missing persons down to a positive identification. We want to be able to, in a sense, really focus in on who this individual could be, and sometimes that is not possible. All individuals who share a particular trait that we are looking at, and fingerprints are a very good example of this, all those who express a particular quality of that feature are included, and those that do not, of course, are excluded. Other kinds of resources that can be used in this inclusion and exclusion kind of process, and this whole process of establishing probability could include things like a body mass index, stature, and sex. So the responsibility for identifying unknown individuals varies quite a bit. It varies by the evidence that you're looking at, and then of course it will also vary by jurisdiction. We know that we are part of a republic in a sense, a democratic republic, and there are, as part of that fabric or that lattice work of different municipalities and different scales of municipal groups, we know that there are different jurisdictions, and in each of those jurisdictions there are different considerations that must be taken in terms of personal and private information. So in most regions, law enforcement is statutorily obligated to establish the identity of a decedent, of an individual who has died, and they also have to, establish a cause of death. Even so, most medical legal offices perform a large number of identifications every year as part of their autopsy and investigation process. Statutes may also stipulate that the identification of unknown decedents is exclusively the responsibility of one group or another, and usually this will fall on the shoulders of the medical legal authority, medical examiner, a coroner. Now, with the introduction of DNA testing as a means of identification, and this really occurred in the mid-1980s, this raised a ton of new issues, not only for medical legal specialists and law enforcement officers, but also people who are concerned about, in this case, personal and private information. DNA technology is most costly. It is very accurate, however, it is potentially contaminated, and as I said, it is costly as it relates to traditional methods. And so this also creates a burden for many of the municipalities that are applying this kind of research to their own identification process. Samples are also an issue. Sometimes they are voluntary, other times they have to be subpoenaed as part of a larger criminal case. Warrants, in a sense, can be sought to forcibly obtain biological samples from suspects, but of course, this usually is not applied to a decedent's family members. Usually that is voluntary in nature. So collecting samples from a decedent's next of kin, for example, may be something that is really outside of the jurisdictional scope of a medical legal authority or a police department, but it is something that can at least be discussed and requested. Let's take a look at different kinds of categories as they relate to the identification process. Standard designations reveal, again, the level of certainty attributed to particular kinds of assessments. We have comparative standards that we use constantly in the field and in the lab. Some of these we have already looked at as part of this lecture series. Generally, these categories of identification and comparison fall into three main types. You have tentative kinds of identification. Tentative indicates that the identity of the decedent is suspected based upon circumstances or any sort of associated artifacts or materials such as wallets, driver's licenses, things like that. Typically, this designation is given to bodies entering the medical legal system prior to any sort of comprehensive establishment of identity. Tentative, in this case, is synonymous with confirmatory or, in this case, directed kinds of identifications. Presumptive identification is a kind of a second category. This meets a higher standard than tentative, yet it is not at the level of a true positive identification. So in this case, circumstances or general characteristics are providing the investigator with a basis for identification. There's no exclusive evidence, however, yet. So for example, you could have, let's say, an elderly white male who is found dead in his home. The door is locked from the inside. There's no signs of any sort of forced entry. The homeowner, in this case, let's say, is estimated to be 85 years of age. He's known to live in this unit or in this location. However, there's no antemortem data available for a definite confirmation of that identification or assessment. So the idea here is that it's presumptive, but it's not completely positive yet. We don't have that one last undeniable piece. There is still the chance for exclusion and inclusion. Now, it should be noted that the medical legal authority can issue a death certificate based upon presumptive identification as long as it satisfies the basis of whatever identification is for that municipality. In terms of what's required by the local system. Positive identifications then represents that ideal class or category, that last one that or third one that we want to associate with this. This is an indication of the individual. This notion of individualization has been demonstrated, and in a sense, you can connect a variety of evidences to the decedent. Fingerprints, nuclear DNA sequences, maybe a medical history, a dental history, of course, if there's skeletal remains or radiographs, connecting all of that. And again, that gives the court system a positive ID on the individual. Now, we hear a lot about DOE designation. What is that? So individuals who enter the medical legal system without any sort of tentative identification are given the designation of DOE until there is a more accurate, maybe even positive identification that can be assigned to the remains. There are no circumstantial clues to provide identity in this case, no missing persons reports, and no basis for an a priori kind of assumption in terms of identification. The term John Doe is actually derived from the 14th century, and again, the British system, in this case, their custom of identifying unknown parties in terms of legal activities and actions as John Doe or Richard Roe, which is kind of a interesting bit of trivial knowledge there. The current legal definition, though, of John Doe is recognized to be a party to legal proceedings whose true name is unknown or withheld. The female equivalent of John Doe is Jane Doe or Mary Major. That's another kind of variation on that. If the sex of the deceased can be determined, the appropriate designation of Jane or John Doe is given to the remains, to the body. All right, let's take a look at, or at least a closer look at, some of the methods that are important to the process of positive identification. And we'll start with this visual recognition. So visible or visual recognition is considered to be really kind of the low-hanging fruit. It's the least reliable form of identification. Despite this fact, it does remain the most common in terms of initial crime scene assessment and any information that's being recorded regarding the decedent. So when a person dies in a hospital or at a home, in this case, or if they're dying accompanied by relatives or friends, or if personal identification is not in question, visual recognition is usually the primary means of establishing the ID of the person. Establishing personal identification becomes significant when an individual dies unexpectedly or away from home or is not known in a community, remains have been found, so on and so forth. And in such cases, visual recognition is based off of photographs or any sort of identifying artifacts, IDs, wallets, things like that. Fingerprinting is another important process when it comes to positive identification. In this case, dactyloscopy is the practice of using fingerprints to identify an individual. Now, fingerprinting today is part of a much larger area of study and research having to do with biometric patterns. Biometric patterns are things that are associated with an individual that relates to their anatomy. And you can look at retinal scans, you can take a look at facial features, you can look at features of the ear, you can take a look, of course, at fingerprints and the patterns associated with each of those. No two individuals, including identical twins, have the same fingerprints, and that's what's important, and that's why early on the FBI recognized fingerprinting and creating a fingerprint database to be important to the identification process. Even superficial injuries can sometimes leave the ridge system unaltered, but things like burns or deep scars and the development of scar tissue can ultimately distort these individualized print systems. And fingerprints are unique to, in this case, primates. All primates express in some way, shape, or form a unique fingerprint. The humans have the most complex. The great apes as well are pretty complex, but the humans have the most complex, and again, they are unique to the individual. Now, latent fingerprints or latent prints are those that are found at a crime scene. And we know that over time, the material that is trapped in the oils that are part of the ridge system that is kind of laid down on a surface, let's say a hard surface, glass or a table, that over time the materials in each of these kind of ridged buildups of material drawn from the skin will begin to break down. It will move into areas between ridges. And you can use things like mass spectroscopy to be able to identify, in this case, particular kinds of compounds as they navigate across a fingerprint to be able to even age the fingerprint itself. So these are pretty complicated and complex resources. The FBI currently stores 250 million sets of fingerprint records as part of a larger database, this integrated automated fingerprint identification system, the IAFIS. And this is what is being connected to and assessed when fingerprints are being recovered. Fingerprints are taken at the time of a suspect's arrest. Fingerprints can also be drawn off of the remains of a decedent. Sometimes, if you have an individual who is of an older age, or the remains have been exposed to the elements for a rather long time, they have partially desiccated, you can actually inject particular kinds of oils and, in this case, rubberized materials and even DI water into the dermis to be able to kind of reinflate the finger to be able to take those fingerprints. But fingerprints are, again, very, very useful in being able to perform some sort of positive identification. DNA testing is another important area. This is a very large subject. We'll just touch upon this for a moment. In this case, in forensic contexts, DNA is going to provide the positive identification of the remains. They'll assign the remains to a known individual. There will be resources that the DNA can be drawn from, blood or semen recovered at the scene. This is also very helpful. And then you can compare, in this case, the recovered DNA to the combined DNA index system, or the CODIS, which was developed by, again, the FBI as a database or a repository for known DNA of known individuals. There are two kinds of DNA that ultimately are assessed, but the more important of the two is the first that I'll mention, which is nuclear DNA. Nuclear DNA analysis for identification relies on, in this case, looking at short repeats or short tandem repeats in the nucleotide of the DNA recovered from the nucleus of the cell itself. And in a sense, this is the most reliable in terms of assigning some sort of biological material to an individual. However, there are problems, and this will come across in court cases. Samples can be degraded, there can be contamination, extragenous kinds of contamination of samples. You can see samples being mixed, sometimes the quantity and quality of the sample, and then the PCR test that is being used, sometimes there are qualities of DNA that can inhibit the process of being able to map out the sequence itself. So there are a lot of challenges, but if you have a fairly useful and tested system, ultimately this is something that can give you some very, very reliable results. Mitochondrial DNA would be the other DNA found in the mitochondria of the cell, the powerhouse of the cell, and there are a variety of methods associated with that. This mitochondrial DNA can give you some major group and population affiliation with regard to the decedent or with regard to the material that is being recovered from a crime scene. The idea of a haplotype and your connection to and your association with a particular haplogroup can be established through a study of mitochondrial DNA. And then we have another method, which is, again, something that is very common to the work of forensic anthropologists, which has to do with forensic odontology and then, of course, osteology as well. Forensic odontology or dentistry uses antemortem and postmortem dental radiographs and records to compare to what is being identified, photographed with regard to a decedent to confirm identification. The same goes for any sort of osteometric data. And then, of course, on a populational level, osteometric data, as you know, can be utilized to establish sex, and age. And then, of course, if there's anything associated with healed bone breaks, different kinds of congenital or well, they would be kind of lifetime related injuries and growth defects. These are all things that can be uh, drawn out from medical records, and you can match measurement to measurement to be able to verify a lot of that information. And in the case with congenital kinds of defects and variations in skeletal remains, you can even establish group affiliation, things like family affiliation and relatedness. So wonderful resources for identification. We will round out this lecture series or this lecture on personal identification with dactyloscopy, and I'll give you a little bit more information and give you some of the common classes of fingerprint patterns and how those are recognized and, in a sense, identified. And these are things that you can even look at on your own finger as you are trying to see where you might fit in the common classes of fingerprints in humans. So again, when we're talking about fingerprints, these are individual and class kinds of phenomena, but all fingerprints are unique to the individuals themselves. When we're talking about fingerprints, we're really talking about the epidermis and the ridges and grooves that are associated with the palm and the fingers. Realize that your palm print is also something that can be utilized for identification. The outer surface of the skin of the palms and the fingers, the soles of the feet, and the toes are all marked by this kind of ridging pattern, ridging and groove pattern. And they appear either as fairly straight lines or as patterned loops and whorls on the tips usually of the digits. And that's why we focus very much on the fingertips themselves. Now, the epidural ridges that we associate with the fingers develop in the third and fourth fetal months of development, as the epidermis is, in a sense, growing and demonstrating or expressing the contours associated with the underlying dermal papillae, which is part of the skin system. The function of the ridges is to increase grip in the hand and the foot by increasing friction and acting like tiny little suction cups. And there's oils and, of course, moisture associated with these as well. And that's why fingerprints are so important to primates, because gripping of the hands and of the feet, this is one of the more important qualities that we associate with arboreal living and, of course, in this case, tree dwelling and movement through the trees. At least that's what the scientific community has consensus on as we currently are. So when you have a smooth object that's being touched, the ridge pattern generally will allow for friction, and it will allow for, in this case, a better grip to be associated with that. Creating friction is always a good thing, at least in terms of the physics of lifting and moving. The epidural grooves on other parts of the skin divide the skin into a variety of areas, and these groove systems are also unique to individuals. The grooves increase in frequency and depth as it relates to regions where we have movement associated with the skin. So if you take a look at the grooves associated with the kind of common divide in your digits, you have the tip of the digit, you have kind of a medial section, and you have a proximal section, that closest to the palm. And if you notice the divide between the palm and that proximal section, the proximal and medial section, and the medial and distal section each have these horizontal, very deep epidural grooves. And then the fingerprint itself on the distal end, the tip, then is all composed of ridges and grooves, and you can distinguish between those. And it's most easiest to distinguish when you have some sort of ink being smeared on that material, or if you have some sort of particulate matter that's being applied to either the latent fingerprint or the finger itself to be able to identify those unique patterns. So when we look at these pattern types, we realize that they can also be classified, and how they behave over time can also be part of that classification process. There are three main pattern types that all humans express in some way, shape, or form. You have arches, you have loops, and you have whorls. And we'll be looking at some of the sub-expressions or subclasses of those categories. Within those categories, there are 8 then sub-patterns or sub-categories, and I'll have you take a closer look at these. And feel free to go ahead and kind of zoom in on some of this. I'll make sure to have a resource to accompany this lecture for you to look at a little more closely as well, to be able to see the various subclasses or categories that are assigned to human fingerprints. There are things like plain arches, tented arches, ulnar loops, so part of that loop group, radial loops. Then we have our whorls, plain whorls, a double loop whorl, a central pocket loop whorl, and then of course we have accidentals or accidental loops. And these all in a sense are, or accidental whorls rather, these all are the kinds of patterns that you will generally see when you're looking at your own finger. In this case, when we're talking about, how these various kinds of prints are expressed, most fingerprints in a human population are in the loop group or the loop pattern. Whorls are much less common and arches are very, very rare. So if you've got an arch, you have a very rare kind of fingerprint. It is unique to you, but there are fewer individuals that have at least that theme of the arch in that case. Finding a latent fingerprint with an arch pattern at a crime scene is valuable for that elimination process that I talked about earlier, because again, it's so rare in the population. Conversely, finding a latent fingerprint with a loop pattern at a crime scene is not as discriminating in terms of being able to exclude or include some of the possible identification pieces to that puzzle. Because again, loops are very common in a population. Pattern types can be used to narrow down the population of fingerprints or eliminate a suspect as a possible contributor to a latent print. And again, the minutia in understanding even unique characteristics, a dot or some sort of oddity in a pattern or a scar, all of these can be helpful identifying resources. All individuals have, again, ridges and grooves. Some of the ridges will end, some of them will bifurcate. Those are all useful when looking at different kinds of identification and things, again, unique to the individual. But again, it is not the sole means of determining personal identification. So take a look at these subclasses. Toggle back to the previous image and I'll include an additional blown up expression of that for you to look at as well, and familiarize yourself with this kind of uniquely human, uniquely primatological pattern that can be used as evidence in the identification process. But the takeaway here is that we're always looking for more than just a single resource or a single source of data. We're looking at everything taken into consideration at the same time to be able to move toward that positive identification as it relates to the work of the medical legal system and, of course, law enforcement.